Next, I'm going to present part of our ongoing research related to Benguela upwelling system in the South-West African coast, near South Africa and Namibia. This schematic here shows the Benguela upwelling system, which is in the north, has bordered with Angola Current, near the border with Angola and Namibia. And in the south has border with here with Angolas Current. As we mentioned, we viewed that nice animation suggested by Stoma. We see that Angolas Current are associated with Angolas rings in forms of mesoscale eddies. Northward Benguelan current bring cold water, and in the subsurface we have poleward current, which brings South Atlantic central water to the Benguela upwelling system. Interesting feature about Benguela upwelling system is that in the north and in the south they are bordered with two warm water from the Angola Current and Indian water in the south. The interesting aspects of all upwelling system, as I mentioned, is that coastal upwelling, they are typically 10 times larger than the offshore upwelling. This is associated with rapid upwelling, which brings high nutrients to the surface and provide favorable environment for large phytoplanktons. They are those phytoplanktons which are good food for anchovies. But at the same time, wind stress curve driven upwelling offshore is associated with slower upwelling and bring less nutrients to the surface, which is favorable for small planktons. They are more limited nutrient environment, adaptable to this environment with limited nutrients, and yeah, they are favorable species for sardines. What's interesting is that catching of sardines and anchovies, most of the time they alternate each other. I mean, increasing in one is concurrent with decreasing of another. What we would like here to address is that how these two types of upwelling related to each other in times of time evolution, whether they follow the same cycle, or they alternate each other, or the positive and negative anomalies, they are concurrent or opposite. To do so, we use different observational data sets. We use ISCAT data set as we discussed at the beginning of this presentation. For the Ekman dynamics, what we need is this theoretical background that has been discussed, and also wind, observational winds. We took ISCAT data sets from 2008 till 2018. Chlorophyll A concentration from Aquamodis was used. Sometimes these data sets shows suspiciously high value of chlorophyll A concentration, which is more than like 30 or 40 milligram per cubic meter. We tag them as miss value, and we didn't consider that in our evaluation. We use ocean model. This is eddy resolving or state-of-the-art ocean model, MOB5, and we force them by observational data sets to bring a real picture of upwelling system. The resolution of this model is high enough to capture mesoscale and partly some mesoscale dynamics, breaking of the internal waves, and so on. The mean state or long-term mean of upwelling is shown in this figure in Benguela upwelling system. As you can see here in the model simulation, which exhibits similar results to the Ekman theory, we can see the pattern shows a strong upwelling near the coast, which reduced toward the sea, reduced seaboard. And we can see different cells here. Cunena cell, Cape Ferrio, Walvis Bay, Luderis, Orange River, Hondeklip Bay, and Cape Columbine. Those are cells that is shown in both analytical Ekman theory and model simulations. Moving from south to the north, we can see kind of northward expansion of Benguela upwelling system, which is captured by model and by analytical theory. It is worth noting that the contours here, which shows the chlorophyll A concentration, shows similar pattern. We can see northward expansion of a region with high primary production. Overall, we can see that on long-term average, Ekman dynamics is working in this system. And by on long-term average having the wind, we can have a good estimate of the upwelling system. When we move to the variation of the upwelling, which is shown in the model, in our model, shown in the left-hand side panel, and in the middle is the analytical theory. We can say, even though the on long-term average, these two show similar patterns, but the variability is different. We see large variations of Walvis Bay and Luderis, which shows large fluctuations in upwelling. And we know that these two are associated with mesoscale and mostly sub-mesoscale eddies and filaments, which cannot capture by Ekman theory. But near the coast, the pattern are more or less similar. Here we can see that, and the same pattern in the variability of the chlorophyll A concentration, but we can see this pattern of upwelling of Luderis and Walvis Bay is missing in chlorophyll A concentration. It is another indication that changing of the physical upwelling is not necessarily associated with biological upwelling. But at the same time, we know that chlorophyll A concentration represent the just ocean surface, but not the primary production in the water column. But overall, the main message of this figure is this variability of Luderis and Walvis Bay and other and overall in the model, which cannot capture by analytical theory. This figure shows the correlation between our analytical theory and the model on the correlation between upwelling. The correlation is very high near the coast, but it is not significant offshore. This indicates that coastal upwelling is a dominant process near the coast. But when we go offshore wind stress curve driven is not the only mechanisms. There are other mechanisms which can play and modulate the wind stress curve driven upwelling significantly and reshape the upwelling. Good example is that of Luderitz and Walvis Bay, where we see large variability. Here we see the correlation is very small. It indicates that mesoscale and sub-mesoscale dynamics plays a major role offshore over this system. When we consider the total amount of upwelling over the entire system, this I forgot to mention that left-hand side panel shows the correlation in each grid cell, but now we integrate the total amount of upwelled water and show that in these three time series. In the top panel here shows the upwelling over the upwelled water over the entire system. The red indicates the coastal upwelling. As you can see the time series here, the time series shows very similar behavior between coastal upwelling, between analytical solution and the model. This correlation retains, but with lesser extents for the blue lines, which shows offshore upwelling, which is wind stress curve driven in the model, and total upwelling, wind stress curve driven in the analytical, Ekman analytical theory, and total upwelling in the model. But still, the correlation is large, is 0.55. It indicates that even though in local scale, the Ekman dynamic offshore is not the dominant contributor to upwelling, but when we integrate over the whole system, the Ekman dynamics is still valid. When we consider northern Benguela upwelling system north of Lüderitz and south of Lüderitz, we can see this for coastal upwelling the same. We can see these two time series are very similar, but it's not the case for offshore upwelling, particularly for northern Benguela upwelling system. For the southern Benguela upwelling system, this correlation is high. It indicates that in southern, in this part, as we can see that large correlation here, when we integrate over the whole domain, Ekman theoretical Ekman upwelling is still valid. This is very interesting results. It indicates that when the, for example, researchers are on the cruise, like researchers like you in different disciplines of ocean science, they are on the cruise and they need to have some estimates of coastal upwelling. We don't need to run the model and wait for 1 month to get the result. We can simply, or we can immediately give you some estimates of upwelling where you are by having just wind and using this theoretical approach. Regarding the correlation between coastal and open ocean upwelling, this figure shows the correlation between offshore upwelling and coastal upwelling at the same latitude. Positive means that or red means that offshore upwelling and coastal upwelling, they are significantly correlated, or we can simply consider they are in the same phase. They peaks and trough at the same time. In the south, we can see that the correlation is positive, which is not the case for the northern sector. It indicates negative, or indicates that coastal and offshore upwelling, they are alternating each other. Positive anomaly of one is associated with negative in another. When we consider this correlation in two different times of the year, this message retains the same. And now for different stations, Cunene, Cape Ferrio, Walvis Bay, Lüderitz, Orange River, Honda-Cliff Bay, Cape Columbine, we compute the correlation between coastal and offshore upwelling. In terms of annual cycle for Cunene, you can see it's very clearly demonstrated here that these two types of upwelling, red means coastal and blue means offshore upwelling, they are in rather opposite phases. Maximum of one is associated with minimum of another. And in this figure, again, when we plot the time series, and this pattern is more similar, we can, for example, positive in one associated with negative of another. It shows that they are frequently in opposite sides. For the Walvis Bay, the opposite appears. The coastal and offshore upwelling, they are in the same phase. And we can see the pattern, we can see they are positive and negative, they are basically in the same phase, indicating that coastal and offshore upwelling events, they are in the same phase or having the same sign most of the time. Annual cycle for Luderitz, we can see for half of the year, they are rather in phase, which is not the case for the for 3 or 4 months of the year, like in September. So, and plotting the time series, the results appear the same. It indicates that the phasing between coastal and offshore upwelling of Luderitz is seasonal dependent. For most of the year, they are basically in the same phase, but for 3 or 4 months, they are in opposite. They are developing in opposite phase. For the Cape Columbine, located in the southernmost, the pattern shows they are most of the time in the same phase. And the correlation between offshore and nearshore upwelling are significant. As we can see here, the shape of annual cycle, they are not exactly at the same phase, but we can consider them most of the time in the same phase. And this is consistent with the result of our analytical theory. Shows that in the south, we're coming back in the south, here, in the south, we see positive correlation between coastal and offshore upwelling. With that, I would like to stop here and summarize my results. In terms of mean upwelling, theoretical model and analytical theory, they agree well, which indicates the dominant role of coastal upwelling associated with divergence of H-band transport. The spatial pattern of chlorophyll A concentration surprisingly compares fairly well with the upwelling in the model and in the analytical theory. We saw that our model results shows extensive submesoscale activities of Luderitz and Walvis Bay. And despite the interruptive impact of these two ocean dynamics, mesoscale and submesoscale ocean dynamics, which we discussed at the beginning of this presentation, model and analytical theory are consistent in terms of total amount of upwelled water in the system. With respect to coastal and offshore upwelling relationship, in the northern sector of Punena and Cape Ferrier, our results indicates that these two types of upwelling are frequently alternating each other. And in contrast, they are in phase of Walvis Bay. The relationships in Luderitz seems to be strongly seasonal dependent. South of Luderitz, again, this relationship is seasonal dependent and varies across the region. This research so far indicates that Benguela upwelling system, when it comes to the biological and physical impact, it is more complicated than we currently thought. With that, I would like to stop here. Yeah, and thank you very much for your attention.
Thank you again, Hadi, for your interesting presentation. That was a lot of data that you showed there. Very interesting results.
Thank you, Natalia. Thank you for your good feedback.
Maybe if no one is raising the hand at the moment, maybe I can go ahead and make the first question, if that's fine. Yeah, I was wondering how did you compute the mixed layer depth? And if you remember what the depth differences are, for example, in Luderitz and Walvis Bay area.
Yeah, this is very interesting question. As we mentioned at the beginning of this presentation, coastal upwelling doesn't mean that we have always the same results in terms of cold water or nutrient flux to the surface. And one of the reasons is mixed layer. Sometimes mixed layer like of Luderitz is deep, takes longer time to interface or density interface to reach the surface. We define mixed layer depth as the depth that the density gradient is small. Is small of some threshold 0.03 or it's basically a result of density contrast in the water. This is what we consider in the model. And the model we use, we are lucky because this parameter is computed by the model itself. Yeah, this is one of the outcome of the model. And we consider, because it is mixed layer, and we consider coastal, we consider upwelling just below the mixed layer, because above that we have mixed. That's why we consider at the base of mixed layer as the estimate of upwelling.
Right, thank you.
Yeah, thank you.
Natalia, there's a question in the chat.
Oh yeah. So it seems that we have a question here from Chris. He asked, Hadi, do you have an explanation for the clear opposite timing of the coastal and offshore upwelling at Kunene?
Yeah. There are several reasons that we discuss about that. One of the reasons, or the main mechanism which can explain large part of this is seasonal cycle. Seasonal cycle in the Bengal upwelling system in the south is overall shows increasing wind speed in summer. But this is opposite for Kunene and Cape Ferrio, that they show enhancement or intensification of wind in winter, in austral winter. But at the same time, the wind stress curve, when we plot the wind stress curve, the annual cycle of wind stress curve, it shows enhancement in summertime, not in winter. And this wind stress curve is mainly associated with zonal contrast of meridional component of the wind, which is maximized or intensifies during summer. For the rest of the upwelling system, like in the south, these two are in phase in terms of annual cycle, but it's not the case in the north. In winter, we have intensification of wind, but a slowdown in wind stress curve. This explains large part of this alternating behavior between these two.
Okay, so it seems that we have another question from Kurt.
Yeah, thank you again, Hadi. My question is the following. Your data are reconstructions of the past, right? So you...
in terms of wind, this is observation from OSCAT scatterometer data sets from satellite. And we force the model with observations, yes.
My question is more, I would like to know predictively, when is the productivity high where? Can you do that with your model?
To best of my knowledge, no, because we use just physics. The model we use at the moment, we have some inputs of biogeochemical cycle, but I cannot tell you a definite statement for that because I haven't analyzed and I'm not aware of that.
But looking at your fingers that move out towards out of to the ocean, couldn't you say if I have a patch of chlorophyll here, which can be observed from the satellite, this will give higher productivity there and there based on your model of Ekman transport?
Yeah, with that, I am a little bit personal. That's why I said at the beginning of my presentation, chlorophyll A concentration is based on ocean surface color. It doesn't show the primary production in the water color. This is one thing. And the second thing is that what we see in the northern part near Lake Kunene, the chlorophyll A concentration is typically large, or of Walvis Bay is large. Even though upwelling is very intensified near Ludericell, but it doesn't show large chlorophyll A concentration from satellite product. I mean, in Walvis Bay, I mean, if we compare the magnitude of upwelling, is definitely maximized in Ludericell. But if we compare the ratio of physical upwelling with biological upwelling of Ludericell, we cannot see this signal of Ludericell. That's why I'm very precautious of saying that. I can tell you about the physical upwelling, but when it comes to biology, there are other mechanisms involved, like the properties of the bottom of shelf bottom water. That at the moment I have very limited information.
I agree with you.
I know that Hadi, as a physicist, loves always these kind of discussions with biologists. So anytime, I would encourage these kind of discussions.
Yeah, and this is quite interesting. That I think if we compare this our knowledge about biology or about the physics, I mean, recently we know that we don't know a lot of things about biology and physics. I mean, knowing unknowns are more in biology than physics. This is my perception so far.
Yeah, because I think it could also have to do with the species composition. Because if you have bloom-forming species in the water, then they're the ones that will probably propagate and keep on reproducing. Whereas maybe Luderitz just has a different species diversity. So some of those species are not able to just, take up the nutrients and, produce readily. So that might be why you have that why they're not linked together, basically, why you don't see the signal.
Okay, yeah, that's interesting. That's very interesting point.
Yeah, that would be very interesting to see what's the recycling of that nutrients. If it's the case that the phytoplankton is not able to take all the nutrients, how efficient they are on recycling, for example, nitrogen or carbon. Yeah, that's a nice topic.
Yeah, so species specific. Yeah, interesting.
Can I say something?
Yes, sure.
Of course.
Dion, yes, from Namibia, and working in the most of the time in the central Namibian part, biology. The biologists from Luderitz might differ. What we've seen, and yes, is that there is some differences in terms of what Chibo said in terms of species, and especially species, the size of the phytoplankton. That it seems that we have smaller phytoplankton in the southern part than in the northern part, and yes, the Luderitz region is the place where it's more quite turbulent. Although I mean, this is the place where you should have a lot of nutrients because of the upwelling. but the system just do not allow the phytoplankton, I think, to that the nutrients are used very effectively at the end of the day. So, and as you go up to the north, things change, as has been shown, and I might also show that in my talks, which is a little bit later. Yeah.
Yeah, that I have one question, Leon. It is associated with because most of the time when the coastal upwelling is strong, at the same time offshore current is also strong. It doesn't let the phytoplankton to stay long enough in the coast, and the retention time is limited. It is can be decay.
Sorry, I couldn't hear the last part of your sentence.
Yeah, sorry. I repeat that. Please, yeah, tell me, yeah, if the voice doesn't have a good quality. My point is that when the most of the time regarding the coastal upwelling, when the upwelling is strong, at the same time offshore transport is strong. Then nutrients, large nutrients come to the surface. It's a nice environment. But at the same time, plankton doesn't have enough time because they moved offshore. I mean, the retention time is limited.
Yes.
Yeah, can be part of the reason.
Yes. I think also sometimes it's also seasonal dependent, that for the nutrients to become available for the phytoplankton, and this is where the physics are playing a role, and you've developed a very strong thermocline. It sort of keeps the nutrients below the thermocline, and it's just allowed when stronger winds occur and the mixing occur, that you will have nutrients coming up in the upper water column, and when things becomes more favorable for the phytoplankton. I mean, yeah, the problem with the information or the data that we have is that we don't have our service is not that regular. So you can't always capture, the whole picture at the end of the day.
Yeah. Yeah, thanks, Don. Nice.
Yeah.
But I think your key is still the species composition. I think that would also give you a good relationship between what you're actually seeing, because that changes everything.
Yeah, thank you.
Yeah, I will be giving some overview next week on the nutrient seasonality we have in our model. I guess that would be also interesting to because we have very limited observational data, and we based most of it on our models. So I guess this will come up again on the discussion.
Hadi, there's a huge ridge in front of Caprio Cuenen in the north, a huge submarine ridge. Why does that not disturb your upwelling measurements and upwelling in general?
In the upwelling system, you mean? Yes.
Yes. It's a very irregular.
I mean, if we if we see the propagation of that in the form of eddies, we see it has some angles and it goes to the middle of the ocean, and it doesn't interrupt the local upwelling that. Of course, there are some interactions, but this is the case that I haven't seen even in the model. Our model, we have some, I should have mentioned that in the southern boundary, we use some observational data sets, some estimates, and we have some uncertainty. That's why the interpretation of our results in very southern part, we should put some cautious on it. because our model suffer from uncertainty from the boundary. But overall, you are right, and yeah, that due to propagation of rings to the Middle Ocean, this part is less affected.